Thus far in the course, we've only dealt with organisms that undergo meiosis correctly or provide a normal meiosis, taking a 2N cell through the meiotic divisions to give us 4N products of meiosis. As with any developmental or biological process, probability for error exists, and one of the errors that we're going to address now is something called chromosome non-disjunction, which we will abbreviate as NDJ. Non-disjunction is a historical term and it's a double negative, so let's just tease out what it means. Junction is just an old-fashioned way of saying joined or together. When chromosomes are synapsed, they are joined. Disjunction simply means when chromosomes separate and are no longer joined. And then non-disjunction now means a failure to separate. And non-disjunction events can take place either in the meiosis I division or the meiosis II division. So we need to start talking about non-disjunction events at meiosis I or possibly non-disjunction events at meiosis II. And what we'll do now is we'll start with the same cell, and here leaving the normal meiosis up as a reference, we'll now start to investigate what these different non-disjunction events will produce, starting with the non-disjunction event at meiosis I. This is where drawing arrows attached to centromeres becomes quite important, because now we're going to have centromeres and chromatids that are attached to them ending up in cells that aren't the normal cells that they would end up in. So normal disjunction, or separation of chromosomes at meiosis I, has one of each homologous centromere being pulled to one of the poles of the cell. A non-disjunction event is simply a case where both copies of these chromosomes end up in one cell and nothing of that particular set ends up in the other cell. So this would produce two cells. Now, this does not mean that there are no chromosomes in this cell. It simply means of this particular pair, there is not a homologous copy of this chromosome in this cell because both of them ended up here. We could conceivably add another chromosome to illustrate this, say big C and little c, and this chromosome pair would just join normally. Non-disjunction is a rare event, so typically it will only affect one chromosome pair at a time, except in the special case we will address in a future video where we actually have an entire chromosome set that fails to disjoin and everything ends up in one cell, and there's specific biological reasons why that can take place. So if we were to add that on here, we would say, okay, that one went there, and that one went there, indicating that this empty set doesn't mean that there are no chromosomes present, just for this particular one that we're interested in. Okay, so the question would be then, well, how do we carry this through to meiosis II? Once the cell is at the stage where it's ready to go through meiosis II, it's not going to make further errors. Again, non-disjunction is a rare event, so rare events happening in multiple steps along the way is unlikely. So we've had one non-disjunction event. It happened during the meiosis I division. Now this cell is going to complete meiosis II to the best of its ability, and what we end up with is as follows. So that's one of the products of meiosis. The other one will be just like it. Okay, and then we'll have two cells that do not have a copy of this chromosome, and as such we would designate them as N-1, N-1 chromosome. And these two up here, I don't have room for it, but we could draw it up here. These are both N+1. One thing that you'll notice about this particular event, which was a non-disjunction at meiosis I, is that it puts non-copied non-sister chromatids together into the same cell. So here are copies of this chromosome. Each one copies the sister chromatids that are copies of each other, and these two distinct chromosomes with two distinct sets of alleles on them. And you'll notice in a non-disjunction event at meiosis 1 is it puts non-sister chromatids into the same cell, and then the n+1 state shows that we actually have non-sister chromatids present as for the n+1 state. So we actually have one of each copy of the 2n set is in one cell. And we also, the other thing to notice is that a non-disjunction event at meiosis 1 produces two cells that are n+1 and two cells that are n-1. And in certain organisms, say haploids with tetrads that you can observe, you can actually make inferences about what's going on and when the non-disjunction event is happening by examining the number of cells in, say, a tetrad that are n-1 or n+1. So in the next little segment, what we'll do is we'll back this up and we'll show a non-disjunction event at meiosis 2. Okay, so here's this same starting point, this cell, which is now we're going to model a non-disjunction event happening at meiosis 2. The important thing to know for meiosis 1 is that if we're going to model a non-disjunction event for meiosis 2, meiosis 1 is going to happen normally. Again, non-disjunction events are rare events, and as such, it would be too improbable to have a non-disjunction event at meiosis 1 and then a second non-disjunction event happening at meiosis 2 because those are independent of each other. So if you're modeling something at a non-disjunction event at meiosis 2, then you should do a proper meiosis at meiosis 1. And similarly, as we saw in the last little clip, if you have a non-disjunction event at meiosis 1, then you model a proper disjoining at meiosis 2. So again, that's why we have our arrows on our centromeres showing proper segregation at meiosis 1. Okay, so there's the results of the meiosis 1 division. And now we're going to take this through to meiosis 2, except we're going to model a non-disjunction event happening here. Now, if we're modeling a non-disjunction event happening in this cell, it is extremely unlikely that this cell will also have a non-disjunction event. So we will model this one normally and this one as the non-disjunction event. You can probably see where this is going and also how it compares to the previous situation. In this case, this top cell, this product of meiosis, will get two copies of this chromosome as before, and this will be n+1. But you'll notice now that these are sister chromatids. They're original copies of the same chromosome that have now been placed into the same cell. We will have an n-1 cell on this side. If this was a tetrad that we could see, then we would notice it in the same half of the tetrad as this n+1 cell. And then we would have two normal n cells, again with the correct disjunction going on here to produce this pattern. So how can you tell a non-disjunction at meiosis 2 apart from the one that we just looked at as a non-disjunction event at meiosis 1? Sister chromatids are in the same n+1 cell. There is only one n-1 and one n+1 cell as a result of this because the non-disjunction event is happening here. And we also have two normal n cells. So whereas in the previous example we had two n+1 and two n-1 cells because of the non-disjunction event at meiosis 1, now we see a different pattern. So what we'll do in the next segment is begin to discuss what happens genetically when you have an organism that has an extra chromosome. So now say if this n+1 product of meiosis fuses with a normal n product of meiosis, this will produce an organism, a diploid, that is 2n+1. So it has a standard diploid set and one extra chromosome. And we'll start to investigate what happens when an organism like this tries to go through meiosis to make products of meiosis.